hemostasis is just a term to describe maintaining the integrity of the blood compartment. That's all it is talking about. And one of the two major processes that are involved with hemostasis is clotting and then being able to break up the clot. Those are the two main things. All right, and so for those, when we talk about the discussion for the clotting homework, hopefully that makes a little bit more sense after you guys did that. Now, what ends up happening is that if you compromise the integrity of that blood compartment, you start exposing the blood compartment to things that it's not normally exposed to. For instance, there's no collagen sticking into the blood compartment, right? You have some cells that make up the blood vessel wall that kind of forms a barrier between the blood compartment and the tissue outside of the wall. But when you compromise the integrity, then structures outside of the blood compartment get exposed to components in the blood compartment, and that sets off a series or a relay of things that happens, one of which is the clotting cascade, right? And some of the other things that happen is that platelets, they start adhering to that area, okay? One of the first things, even before that happens right away, is that you kind of get something of a contraction, try to minimize blood flow to that area so that the blood doesn't leak out from that compromised integrity of, whatever structure's been damaged. Now, like we talked about in the quiz, this whole idea of platelet aggregation is a positive feedback process. So a few platelets, they're adhering to that area where you have blood vessel wall damage, and they are getting activated, sending out factors to recruit more platelets that then get activated, that send out more factors to recruit more platelets, and so you get this huge event that goes on with platelet recruitment and activation of platelets, right? And so one of the major formed elements that you find in clots are going to be platelets or thrombocytes. Okay, does that make sense to folks? So some of the chemicals being released are up here. One that's really potent is this platelet-derived growth factor. That's what PDGF stands for, and it's fairly potent. And there's versions of it that sometimes are used to promote clotting, but calcium's important for the clotting, as we'll see, and then ADP, hopefully people that understand what ADP is, right? And there's other factors that get released that activate platelets and help promote this clotting event that goes on. Make sense to folks? All right? Yeah. Yeah, platelet-derived growth factor. Mm-hmm. All right. So this is pretty much kind of what I was just telling you. You get vessel damage, and the specialized epithelial cells are called endothelial cells that line blood vessels. And so the endothelial cells will release chemicals, but you also get components underneath the endothelium getting exposed to the blood, tissue, and therefore going to activate and recruit other platelets. And then vascular spasm, that's kind of the contraction that goes on to try to minimize blood flow to that area, to minimize blood loss. And then we have this positive feedback, where platelets attach, they get activated, and they recruit more platelets, right? And then more platelets get activated, and so forth. So that's kind of the start of this process. Now, there's proteins, right? And they're really enzymes that are floating around in our blood, and they float around mostly in the inactive state, and those are the clotting factors, and you don't have to memorize all of them. There's a whole series of them. What ends up happening is that there are certain triggers that start this cascade or series of clotting factors to get activated, ultimately leading to a mesh that forms to help form the clot. Now, one of the triggers is this tissue damage. The other one is you can get activated certain activated factors coming from platelets, right? They all these triggers funnel into activating this factor 10. That's one of the few that you probably should know. Factor 10 is a really important one, and that is going to lead to the other ones you need to know, where we go from prothrombin into, prothrombin activator, which converts prothrombin into thrombin, and then thrombin converts fibrinogen into fibrin. And then the fibrin comes together to form a clot. Now, I posted some really good animations of this process up on Blackboard. Puts things in motion and makes things a lot more sense rather than talking about it individually. So please take a look at that. But I want to focus in on kind of this area here, right? The whole point is to convert this fibrinogen into fibrin, which then can form this meshwork to hold the clot in place. We don't want to have fibrin floating around, though, because then our blood would be clotting all the time, right? That's a bad situation. So what do we have? Well, it's manufactured as a inactive protein, okay? Right? So it's manufactured as fibrinogen. Now, fibrinogen gets converted to fibrin by thrombin. So if we have thrombin floating around in our blood, right, it's going to automatically convert the fibrinogen into fibrin, and then we'd have that same problem of clotting going on, right? Makes sense. So rather than having thrombin being manufactured as an active form, we have it manufactured as an inactive form, prothrombin. Okay? Now, there's a prothrombin activator which converts that prothrombin into thrombin. So here, we don't want to set this series of events into play, well, when our bodies don't want to, because then we have clotting going on all the time. So here, this prothrombin activator gets activated by factor 10 being activated. Factor 10 only gets activated if we have this series of events or this series of events, usually both of them, right? So that's what I'm talking about, where you have this whole cascade of events, right? Now, in order for this cascade of events, this clotting to go on, only when it's appropriate, a lot of these clotting factors are manufactured in their inactive state, and then they get converted to the active state in the area and at the time when they need to. Everybody with me on this? Okay? All right, so all right, so this isn't the last time we're going to see this, so I want to introduce this concept. Whenever the body is manufacturing something, right, and it doesn't want it to be active right after it's manufactured. It wants to make sure it stays in an inactive state and only gets activated when and where it needs to be activated, right? Okay, so this is going to happen, say, in the digestive tract, right? When we want to break down things inside of our intestine, but we don't want to break down, say, our intestine, right? Okay, so what does your body do? Well, it makes inactive versions of it so that it becomes active only when and where it needs to be active. Same thing with these clotting factors. Now, one of the terms that's used is something called a zymogen. So zymogen is basically an inactive protein precursor that becomes activated, right? Usually by an enzymatic reaction by another protein. Okay? Like I said, clotting factors, so you can classify them as precursors or zymogens. Not the last time we're going to see this, though. Okay? Now, if we think about it, so let's say that we'll talk about prothrombin here. Okay? Right? So prothrombin is the inactive version of thrombin, let's say. Okay? It's an enzyme. All right? So if it's the inactive version, we could call it a zymogen. So prothrombin. So how does it work? Well, it turns out enzymes, they usually need to have their substrates, whatever they are going to be catalyzing, helping catalyze, they need to have access to the active site of the enzyme. A lot of times what happens is the inactive protein has a part of it that inhibits it. It kind of sits in that spot so that the substrates can't get to it. It's kind of like parking in the parking place, right? And so as long as that is parked in the parking place for that enzyme is not active. Everybody with me on this? Now, if you cleave, meaning that you proteolyze, you remove that inactive or that inhibitory part of the protein, now that active site can get to or can find substrate and catalyze a reaction, right? So that's what I'm talking about here, that we have an inhibitory part of the zymogen that's blocking the active enzyme part of it, okay? Right? So this would be inactive. Now, most proteins, they fold in all sorts of funky shapes, and so I kind of drew it out over here, right? Here's our active part of our enzyme, and we're going to call this area in here the active site, and then the red part is representing the inactive portion of it, right? So here's our kind of our active site in here, and it's being blocked by the inhibitory part of the protein, okay? Right? And so we could say that, you know, this is prothrombin, okay? And then if we wanted to make this active, then we would basically chop this off, right? And then we would be left with our enzyme that's now active that we can call, say, thrombin. Okay? Everybody with me on that? And so here, what does this cleaving? Usually it's another enzyme, right? It's the activating factor, prothrombin activating factor. Yep, and then what activates prothrombin activating factor is factor 10. Yep, exactly. So it's this cascade, this relay of events that goes on. We're kind of starting in the middle a bit, you know, with looking at prothrombin to thrombin. So here, this is our inhibitory portion, and then if we cleave it, right, then this is going to be removed and we'll be left with the active form of that. Everybody with me on that? Okay? So here, what ends up happening is that if we get rid of the inhibitory portion, we're left with the active form of thrombin, right? And then thrombin catalyzes the conversion of what to what? Fibrinogen to fibrin. So fibrinogen and fibrin are also zymogens, right? So here we could talk about another protein that is downstream of this, where we're going to be converting, let's see, how do I want to draw this? Right. And so we could call this fibrinogen, right, with the inhibitory portion of it. So right? Everybody with me so far on this? Okay. And what does, say, this is activated thrombin now? What does this do? What it cleaves a part of that fibrinogen off so that now the fibrinogen starts forming fibers, right? Forms a meshwork. So here, this is going to be converted into active form of fibrin, but only when thrombin is active. And thrombin's only active if we cleave that inhibitory portion of prothrombin to make thrombin. Everybody with me on that? So it's a series of events to basically activate the inactive zymogens, right? So that we don't get undue clotting. Now, some people have problems, right, where this can be problematic, right? And so here, once fibrin's been activated, we could get multiple fibrins to come together to form that fiber, that meshwork that encases a clot, and it basically is enclosing some formed elements. Does that make sense to folks? All right. It's a great animation that I put up on Blackboard that kind of talks about this a bit. Okay? Now, like Sean was getting at, what does the conversion of prothrombin to thrombin? Is that prothrombin activator? What converts the prothrombin activator is activation of factor 10, and then we have a whole series of events that are important to get to that activated factor 10, though, right? So this is a tightly regulated cascade. The other thing I want to say about this is that calcium's important. Calcium's important. And so here, if we don't have calcium, we have problems with clotting, right? And in order for some of these proteins, in order for them to use the calcium effectively, they need access to vitamin K. So if you interfere with vitamin K, you interfere with the ability to clot. Now, how can you reverse that? Well, you can give vitamin K, right? Everybody following me on that? Okay, hold on. Vitamin K. It's important for making sure some of these factors in the clotting cascade work effectively. All right? And so if we don't have enough vitamin K, or if we interfere with vitamin K, then these clotting factors don't work effectively. How can we reverse that? Well, we add vitamin K. Sorry, go ahead. Platelets are budded off of megakaryocytes. These clotting factors are coming from other parts of the body, but platelets are important because they release factors that promote clotting, the clotting cascade, as well as recruiting more platelets. Yes, that's their main role. Everybody following me on this? Okay. All right, so let me just finish up real quick, and then we'll have our discussion here. All right, so fibrinogen gets converted to fibrin, and that's done by thrombin after it's been converted from prothrombin, and then this is helping lead to the clot, right? And so you can see a lot of formed elements that are caught in this meshwork of fibrin. Okay, now clotting cascade drugs, you guys are probably going to run into these if you go into healthcare field. You don't have to memorize this whole series of events, but I just wanted to point out where they work. How many folks have heard of heparin? Right, so heparin is commonly used as what? So it promotes clotting, or is it preventing clotting? So, yeah, so it's preventing clotting, right? And so here it's interfering with certain factors, right, in the clotting cascade. All right? So warfarin, how many folks have heard of warfarin? So blood thinning medication, also a rat poison, right? So it has a very narrow therapeutic index, meaning that you have to monitor patients' levels of this really closely. It's also known as Coumadin. Yep. And so here, that is impacting certain factors as well. So this interferes with that vitamin K pathway that I was telling you about, right? So individuals that have too much warfarin on board, right? What's happening? Are they promoting too many clots, or are they having problems forming clots? Forming clots, right? So if they get a subdural hematoma, so what ends up happening? We're not able to form a clot. What happens to the blood accumulation? Keeps accumulating, right? We've basically not able to maintain hemostasis. What do we give to try to counter that? Vitamin K, right? Because warfarin interferes with the ability of vitamin K to help maintain or make it active forms of some of these clotting factors, right? So those are a few that I just wanted to point out. Now, down here is, so here we've got fibrinogen to fibrin, right? That's the end of our clotting cascade. Everything down here is about breaking up the clot, okay? So over here in the lower right-hand corner is everything that's involved with breaking up the clot, okay? And so there's certain, pharmaceuticals or treatments that interfere with this pathway as well, okay? But those were a few I just wanted to point out. So you don't have to memorize everything on here, but I think it's handy to know that there's some important compounds that patients that are you're probably going to see if you go into the healthcare field are going to be on. All right? Make sense to folks? Don't memorize this. Just know a few of the major details that we just talked about, right? Warfarin, heparin, and so forth. Very clear? You guys going to memorize this? Yes. All right. Okay. Yeah, so fibrinolysis, we talked about how clots form, but we don't want them around forever, right? So what do we want to do? We want to break up clots when everything's repaired. So how does that happen? Well, we're going to have our fibrin, and we want to break up the fibrin. Well, that's the job of plasmin. We want plasmin around while we're trying to make a clot? Probably not, because as soon as we start making fibrin, plasmin is hydrolyzing it, breaking it up. So we only want to make plasmin, right, when we need it. So how do we do that? Well, we once again use that strategy of zymogens. So we have plasminogen that can be converted into plasmin. Well, plasminogen only gets converted into plasmin when it gets converted by tissue plasminogen activator or uroplasminogen activators, two forms, two areas that plasminogen activator comes from. Okay? All right, does that make sense to folks? All right, so everybody kind of clear on clot formation and then breaking up the clot? All right, so last thing you want to do is have these things active when they're not supposed to be active. So the zymogen strategy helps, right? Zymogens, people, you don't have to be like 100% comfortable, but is it kind of understandable? Yeah, so you might have to, think about it a little bit, right? All right, so what we'll do, right? So here's a bit of a review there of plasma components and functions. And then this is a review of taking a look at the components of the blood, including what we just talked about with, on the quiz with formed elements versus what we find in plasma. Okay? Right? Okay. Right? And this is review slides. So what we'll do now is transition into just having discussion. Why don't we take a 5-minute break, and then after the break we'll have our discussion of the clotting related homework. So does everybody understand what the cardiac cycle is about? So what's the cardiac cycle? So cycle that blood takes from the heart to the heart, through the body. Yes. So here, this is really important because students always get confused between the cardiac cycle and the cardiac conduction system. So you're going to want to pay attention to this. The cardiac cycle is the physical movement of blood through the heart. Cardiac conduction system, just like if you're thinking about conducting electricity, is the electrical components. And remember, the heart is basically a muscle, and in order for this muscle to be stimulated to contract in a very specific sequence, it has to have a very specific sequence of electrical events that go on, that precede each of the parts of the contraction in the cardiac cycle. And what you guys are going to be doing in lab this week is taking a look at the cardiac conduction system. You guys are going to be looking at ECG. We're going to have Dr. Jack McKenzie come in and help you guys out with that, who's a cardiac physiologist who knows a lot about the ECG and cardiac conduction. All right? And so here we're talking about filling atria and then ejecting blood, or filling atria, filling ventricles, and then ejecting from atria, injecting from ventricles, right? The physical movement of blood. It happens because of the rhythmic contracting first of the atria, then of the ventricles, right? What stimulates these contractions is the electrical signal, the cardiac conduction system. What you want to know as a student is how the cardiac conduction system and the cardiac cycle, right, relate to each other. I can guarantee you there's going to be a question or two on exams about that, right? So when you talk about the cardiac conduction system, how is that related to events in the cardiac cycle? What parts of the cardiac conduction system are causing, say, the atrials to contract? What part of the cardiac conduction system is stimulating the ventricles to contract? You want to be able to relate what's happening in the cardiac cycle to what's happening in the cardiac conduction, cardiac conduction system to what's happening in the cardiac cycle, right? And back and forth. It's really critical to know how these two processes relate to each other. Now, is everybody familiar with the term systole and diastole? So what is systole? So when we talk about the atria are in systole, so what are we talking about there? So atria is pushing blood to the ventricles. Contraction and relaxation. Systole is squeezing. Diastole is relaxing. So we have rhythmic squeezing, relaxing, squeezing, relaxing. The heart is a pump, right? So if you haven't taken a look at the background of the heart and the cardiac conduction system lecture that I posted, you need to do that. You should have done it already, but if you haven't done it, you need to do that as soon as possible. One of the things I want to make sure that you're familiar with, because we're going to revisit this, right, is this idea of stroke volume. So normally, right, when we're talking about how much blood is ejected from the heart, we're more interested in blood being ejected from the left ventricle. Why is that? So it's the more oxygenated blood. Where's that blood going? To the body, systemically, right? So here, when we talk about stroke volume, we're generally talking about how much blood is being ejected from the left ventricle. Okay? Now, it turns out the reason I have this up here is because it does a nice job of talking about how we measure stroke volume in the left ventricle specifically. All right? Okay? So here, when we have contraction going on, or before we have contraction, systole of the ventricles, the left ventricle in particular. That ventricle is filled with blood, right? Okay? after it is gone through systole, right, there's still some blood left in the ventricle. So here we start off with this amount of blood, okay, and then we're left with this amount of blood in the ventricle. Can we figure out how much got ejected? How much was pumped out of the ventricle? Yeah, absolutely. How can we do that? By subtraction, right? We start off with this amount, we're left with this amount. The difference between the two is how much got ejected from that ventricle. That is called the stroke volume, okay? So the total amount we have in the ventricle before squeezing, systole, right? Before systole, when it's in relaxation, right, is end diastolic volume, right? Total amount in, right? Okay? Make sense to folks? The amount of blood left in the ventricle after systole is end systolic volume, right? You subtract the two, you figure out how much blood was ejected from that ventricle. That's called the stroke volume. Everybody with me on this? Okay? Because this is going to be really important to talk about when we talk about, say, blood pressure. Okay? Because if we start mucking around with end diastolic volume or end systolic volume, we're mucking around with stroke volume, and then we're impacting potentially blood pressure, right? Okay? So I'm setting you guys up for a little bit later discussion about this. Does that make sense to folks? Feeling good about it? All right? For those that haven't seen this lecture yet, you need to take a look at it soon. Okay? All right, so I'll ask again, do you guys have any questions on the cardiac cycle? Okay. All right, so I'm just going to give a brief intro to the cardiac conduction system so that you guys are going to know a little bit of what's going on in lab. Plus, when you guys are taking a look at the slides, then maybe hopefully it'll make a little bit more sense to you. Remember, this is the electrical component of the heart. It's not the physical movement of blood. That's the cardiac cycle that we just mentioned. Okay? But in order for muscles to contract, they need to have a electrical stimulus, right? So this electrical stimulus is promoting the sequence of contractions that we know as being part of the cardiac cycle. So they're intimately related, right? Now, two parts of the heart that are really important to know. First of all, the heart as a tissue, it doesn't get stimulation from the nervous system. It has its own rhythmic cells in there. They're called pacemaker cells. So it doesn't need to have the central nervous system constantly telling it to contract. Right? And so there's two areas that kind of have these, rhythmic electrical impulses that stimulate the rest of the cardiac tissue to contract. And the cardiac tissue is different than skeletal muscle tissue in that they are connected to each other, not just, physically connected to each other, but the cytoplasms between cardiac muscle cells can communicate. So when one is stimulated to contract, its neighbors are also stimulated to contract. So they contract as a unit. Okay? All right. So here, one node, the sinoatrial node, is located up here in the right atrium. Sinoatrial node. This is where electrical impulse starts out and it spreads. It's going to spread down through the right atrium, and then it's going to spread over towards the left atrium. Okay? This is the electrical impulse. Now, there's another node that is going to receive that stimulus called the atrioventricular node, the AV node. Okay? So here, this electrical stimulus spreads from the sinoatrial node across all the left and right atria, and it gets to the AV node and it pauses for a millisecond, right? A fraction of a millisecond. Okay? Now remember, this electrical impulse, what it's doing is it's stimulating that cardiac muscle to contract. So first, we're squeezing up here to push blood into the ventricles, and then eventually we need to squeeze the ventricles to eject blood from the ventricles out to systemic tissues, or in the case of the right ventricle, to the lungs. Okay? So in order for it to make sure that the blood gets squeezed in the ventricles, there's a bit of a pause before the ventricles contract. Right? We squeeze the atria, and there's a bit of a pause in the electrical impulse to allow the ventricles to fill with blood before they contract. That pause happens there at the AV node. Okay? So the AV node then is sending that electrical impulse after the pause through the interventricular septum, and then it branches, right, into surrounding cardiac muscle tissue to stimulate the ventricles to contract. This whole series of beats per minute, or this rhythm, at the SA node, the sinus rhythm, is generally about 60 to 100 beats per minute. How many people will have a heart rate of 100 beats per minute? Probably not resting, right? We'll find out why in just in a little bit, probably not today. Nodal rhythm is about 40 to 50, so a little bit slower. Okay? If you have something called an ectopic focus, this is basically where some cardiac muscle tissue is spontaneously firing. So it's somehow like some tissue over here suddenly depolarized to cause an electrical signal coming from an area other than the SA and AV node. Is that a problem? You bet it is. Right? So that's an ectopic focus. Okay? So here, this is what I just told you guys about. This initiates the heartbeat, sends an impulse over the atria, AV node receives it, pauses a slight fraction of a millisecond, and then sends the impulse through to the ventricles, through the atrioventricular bundle, and then there's fibers that branch out called Purkinje fibers that come out here that are going to be propagating that signal to the surrounding cardiac muscle tissue, right? This is the basics of the cardiac conduction system, right? Now, don't forget how we talked about electrical signals in nervous tissue, right? Where we had voltage-gated channels, we had depolarization, repolarization, right? We had to reach threshold in order to get an action potential. Same rules are going to apply here. So if you are uncomfortable with that, be sure to review that. All right, so I think I'll leave it there, and we'll pick up the discussion on Wednesday about this. And be sure to take a look at the lab before coming to lab. All right? So be sure to turn in your homework before you leave.